Let me show you homework that one of the previous students submitted before. This student proposed to investigate the strongest DNA, which is the genes that are highly resistant to external pressures. I liked this student's idea to explore the strongest DNA. This student suggests that samples of many different synthetic DNA molecules be exposed to diverse harsh environmental conditions such as intense lighting, high temperature, the heating, and acidic conditions or basic conditions. The surviving molecules can be selected, and you have now discovered the sequence of strongest DNA. Such a sequence may be identified in the genes of particular living organisms. That's a great idea. However, there are a couple of potential problems. One is that the selected DNA molecules may be rare and may be hard to detect. You need to prepare a numerous number of DNA samples one by one. For this reason, my recommendation is to use a mixture of randomized DNA molecules as a starting point and to use PCR to amplify the selected DNA molecules. So if the defined short sequences are placed on both ends of randomized DNA molecules, like this, the DNA molecules that will be selected eventually can be amplified by PCR reaction using the synthetic DNA primers complementary to the defined sequence. So then once you select such a gene, you can do the PCR amplification using the primer that are complementary to these ends. The amplified DNA molecules will also be exposed to the harsh environmental conditions again. If some of the DNA molecules survive, they will be amplified again through PCR reaction. This amplification and selection process will be repeated 10 times or more. Then this process should eventually yield the strongest DNA molecule. Actually, such experiments have been done. In the real experiments, RNA is usually used instead of DNA. Why is that? The DNA has a double helical structure. As you learned in this course. But on the other hand, single-stranded RNA molecules are capable of forming different shapes and patterns so that RNA molecules are more likely to have diverse functions. So this is DNA. The RNA molecule would be like this. In the next video clip, I will show you the real technology to select RNA molecules that have particular functions.